I think we can maybe, yes, it's 12:03. I think we can start. Huh? Okay, so hello to everybody and welcome to this monthly webinar of EUROTOX. Today we are lucky to have the Professor Olivier Taboureau that is going to give us a presentation on machine learning methods applied to toxicology with biological data. And he will give example with transcriptomics and cell morphology readouts. So Olivier he has a background in biochemistry. He obtained his PhD in 2001 on bioinformatics and data mining in France. Then he moved to industry in Denmark, in Novo Nordisk, when he studied antimicrobial peptide and coagulation factor using QSAR and molecular modeling approaches. In 2005, he joined the Center for Biological Sequences Analysis at the Technical University of Denmark. And then he built his group on computational chemical biology. And in 1210, he came back to France and then became a professor at the University of Paris Diderot. Now it's Paris Cité. And he started to work on computational pharmacological profiling and developed a web server, ChemProt, to mine chemical biological data. Since 2018, he is a member of the computational modeling of protein-ligand interaction team at the biological and functional unit at Université Paris Cité. With his expertise in bioinformatics and chemoinformatics, he is contributing to the development of computational approaches related to pharmacology, drug safety, and chemical risk assessment. And of course, he has a lot of interest in the application of system pharmacology and system toxicology with the analysis of omics, transcriptomics, and high-content imaging data. So I'm very pleased to have Olivier today. I think it was timely to have a presentation on all the approach concerning machine learning methods in a EUROTOX webinar. And so, as you remember, the people that are used to come to the webinar, we have a 45 minutes presentation, and it is followed by 50 minutes of question. And so you can put your question in the chat, and then if you have sometime more, we will ask questions directly. Okay, thank you, Olivier. The floor is yours.
Okay, thank you for the introduction. So just to be sure that everybody will be on the same page, I will focus essentially on one technology, which is QSAR, and how we can apply, some kind of machine learning approach used in QSAR, but there with transcriptomics and cell morphology readout. So everything related to molecular modeling, docking, will not be present on this talk, but of course we are doing also such kind of approach and might be come to another discussion in another webinar. Anyhow, so can I pass my next slide? Yes. So I'm Olivier Taboureau, professor at the University of Paris Cité, which is located in Paris, in the center of Paris. And I'm part of a unit which is co-biology functional et adaptative, where people are essentially a wet lab. So they are making experiments on different domains, like in obesity, in cancer, in toxicology. And I'm part on a unit which is called CMPLE, for computational modeling of protein ligand interaction. And here we are only bioinformatician, chemoinformatician, so dry lab in some way, where we are around 30 people working only on computer. And the main objective is really to understand relationship that we can have between molecules, any kind of molecules that can be environmental chemicals, drug, natural product, but also peptide and RNA. And also connection with proteins or genes, or also sometimes tissue, depending on the level of information we have and complexity. So, as I said, we have many experience in bioinformatics, chemoinformatics. My main focus is really to try to, from a molecule to a clinical effect, which can be beneficial or which can be toxicity or reaching to some adverse outcomes, try to understand, I mean, at the molecular level and at the cellular level, what can contribute to the observation, the phenotypes that you can see on some clinical effect. And for that, so we integrate different type of information, chemogenomics, for example, where we are looking on the impact of a molecule on a protein directly, but also looking on gene expression to see, I mean, how it can perturb a full ensemble of genes at the cell level and at the pathway also. And we are looking also on some genetic variation, how it can impact also some susceptibility for some individual that can, I mean, also induce some reaction, some adverse outcome reaction when they take a pill. But today we will talk about toxicology and exposome and how we can use some bioinformatic and chemoinformatic tools to make some prediction about risk that can be associated to this exposure of chemical. And we know, I mean, we are really exposed to many kind of chemicals from the air when we are taking drugs, from the food as well, from water. And then I show in a review, which is basically 5-10 years ago now, that's basically, I mean, all this chemical exposure, I mean, can lead to close to 430,000 deaths per year. And also, I mean, can also lead to some adverse drug reaction. When I put, I mean, close to 200,000 adverse drug reaction, it's really severe adverse drug reaction. It's not only a rash or maybe you can have a headache, but that's severe, that can really come to death. And so it has been also presented that worthwhile we can reach close to 5 million of environmental exposure deaths per year due to the exposure to different chemicals. The problem that's if we want to make in advance some estimation, some assessment about the risk related to chemicals, and we want to do that experimentally, we know it's costly. It requires some animal testing, and it can also be complicated to test many compounds at different time, at different dose, on different species, different conditions. And that's the reason why, I mean, people are trying also to shift a little bit all these experiments that can be done, I mean, in vitro or in vivo, through, I mean, some computational tools that might help or at least prioritize some chemical risk and to make some assessments in advance before to really do some experiments in animal and even in human. But then that's a question that usually people ask us. It's can we predict, I mean, chemical toxicity using computational approach? And the answer will be in some time yes, in some time no. So like that, you can don't have any expectation from my side, but at least we can guide a little bit the agency, I mean the scientist about potential risk for some chemicals versus, I mean, more safe chemicals. And it's really depending of the data that we have in our hand and the diversity also of the structure of these compounds that can help also us to make some good models or more limited models for predictions. One technique that we use a lot and people are using a lot, it's called, I mean, quantitative structure-activity relationship. It's used in pharmaceutics, in cosmetics, also for biocide, pesticide prediction and reach, where the idea is really to connect some biological endpoint or toxicity endpoint with the structure of the chemicals. And through that, I mean, there are some reliable and accurate QSAR that can be performed. So the principle is in some way simple, where basically the general idea is to develop a mathematical models using a function, a mathematical function, which are able, to relate the biological endpoint with some parameters which will be related to the structure of the chemicals, and using, some biological toxicity which can be continuous. You are looking at IC50, LD50, for example, of molecules. It can be a class, saying if it's toxic or not toxic. And then you have a list of what we call molecular descriptors, which is connected to the structure of a compounds. And again, on this structure of a compounds, on this list of descriptors, you can have quite simple ones, which will be related to just the weight, the size of the molecules, to a more complex one where you can take the conformation in consideration, the energy also of the molecules, some connectivity with different atoms. And so you can encode them through what we call fingerprint or descriptors, which will give a long list of information for each molecules. And at the end, you will get something that looks like that, which is a matrix. For each compound, you will have a toxicity endpoint or a biological endpoint information, which can be continuous, 55, 8, 33, 22. Or you can use a threshold to define, below 20, for example, it starts to be a toxic molecules. And then you will use a threshold to define toxic or non-toxic to make not anymore a regression model, but a classification model. And that's the same for the molecules. The molecular features can be defined with a continuous value, like the molecular weight, as I said, but it can be also encoded by a binary information. Do I have a presence of a ring system or not? If yes, I put a 1. If not, I put a 0. And this matrix will be, the basis to make our QSAR models, because that will be with a mathematical function, which can be linear or non-linear, that you will be able to build a QSAR model and make some prediction using some statistic to define, the accuracy of your model or the performance, the robustness of your model with a R-square or a R for a regression. And for classification, we're using more a balanced accuracy, sensitivity, specificity to check, the reliability of your model, and to define, if you can use this model really to do some prediction which will be accurate or not. So that's the principle of QSAR, using essentially the structure of a molecule, and that people are applying in different domains. Here, it's a review I found, on the scientific reports, where basically you can have a list of tools, web server, which then so you can play with them, and they will give you some prediction about some rodent oral toxicity, about endocrine disruption, about if a compound can be a hair blocker or not, about skin sensitization. So all these tools are available for you online, and you can play to do some prediction. All of them are based on the structure of the molecules, and so you need to have at least the structure of your molecules to be able to do some prediction of this molecule about these different toxicity endpoint. In our teams, we are playing with the same machine learning tools, but instead of using the structures, we wonder if we can use the biological response of your systems or rat or another species or on the cell, and to see, how they can respond when the cell is exposed to a compounds. And that means we can use not anymore the structure of the molecule, but really a response of your organism in presence of the molecules. And to do that, we use, for example, data which is coming from toxicogenomics. So toxicogenomics is basically something that you do in lab, where you can study the response of a genomes to what we call environmental stressors and toxicants. So you have a cell line or you have a tissue, and you expose this cell line to a molecule in a specific concentration and for a specific time. And for that, you will get at the end a list of genes with their perturbation, I will say, in presence of the molecule. And so you can make a comparison of how much will be the deregulation of your genes or set of genes in presence of the molecule compared to if the molecule is not present, I mean the cell line as it is. And so like that, you can in some way identify, I mean, what we call useful biomarker, which can be related to some toxicity endpoint. If you have many compounds that you know they are related to some liver injury, we will see some example after. Do they share in common what we call a gene signatures, so a set of genes which are deregulated in the same way. And so like that, we can suggest some biomarkers that seems to be related to the biological or the toxicological endpoints related to a set of molecules. And so it can, like that, elucidate what we call the molecular mechanism of toxicity of a molecules, considering, I mean, the biological response, the gene response from the cells when it's exposed to molecules. So to get, I mean, the information, so you basically have, as I said, I mean, the cell lines where, I mean, you can prepare the RNA because it's usually based on RNA. And then you have different way of quantifying, I mean, the transcript, which might be related to some chips array. That's what they use, for example, in a technology called L1000. Or they can use directly some high-throughput sequencing, which is a more modern way of quantifying the transcripts, where I mean you have small sequence of your genes, and you can also measure the variability of the response of these transcripts when they are in presence of chemicals versus when they are not. And so like that, you will get, like for QSAR, a matrix with for each line a compounds annotated for a toxicity endpoint of interest on your study. And all column will be, I mean, basically the genes deregulation that you can observe when a cell and all these genes are exposed to a molecules. So the matrix will be very large because you can get, let's say, 1,000, 5,000, up to 30, 50,000 of molecules with, for each of them, I mean, between 10,000 and 30,000 of genes when you can see the deregulation in presence or in absence of compounds. And as it is a matrix like QSAR, then you can start to be able to do some machine learning application and to make some models that could be used to do some prediction for new compounds for which you have also a transcriptomes profile. You need that. So it needs to be performed on lab, on a wet lab. And then, I mean, if you have the list of transcripts for a compound for new compounds, then you can develop or you can make some prediction with the model that has been developed for the compound of interest. So it's an example of where you can get such kind of toxicogenomics data. Well-known, it's called TGGATE, that has been developed in Japan, and basically for 170 compounds, they managed to get some transcriptomes for rat hepatocyte in vitro, human hepatocyte in vitro, in different dose, in different time, and also in vivo in rats, where, I mean, they could get some slides from kidney and livers and do also some transcriptomics analysis on it. So the tables, basically for each compound, that's what you will get. A list of genes, a log for change that will measure, the deregulation of the genes in presence of a compound, which is here, the Y14643, and a p-value where, you can measure if it's already a significant change compared to the other genes or not. And so, like that, for each genes, when you see a high log4 change, it means a high perturbation or deregulation of your systems. You have also information about on which pathway these genes are also involved. And for example, the ACOX1 is related to some lipid metabolism. So you can make some assumption that if you have a high deregulation of these genes in liver, you might impact the lipid metabolism, and it makes sense if it's in liver because we know it's a place where also you have a lot of metabolization. And here's another example with the transporter SLC27A2, where again you see a upregulation when it's positive, so you have a deregulation which is higher compared to the normal of the genes. And so you can see it's related to some fatty acid transport. So you will have a higher regulation of these genes in presence of the compounds. And when you have a minus, it means then you have a deregulation. So you will reduce the function of these genes when it's in presence of molecules compared to when it's not. So like that, you have a matrix. You have a biological endpoint for some of the compounds. You can be able to develop kind of QSAR methods, but with gene expression data instead of using the structures. And that's what we did in 2020 with what we call drug-induced liver injury compounds, where for basically 190 drugs that has been tested on 2 cell lines, which were, MCF7 and PC3. It was a challenge developed by CAMTA to 2018. The plan was to try to develop models that are able to predict if a compounds might, induce some liver injury or not using transcriptomes information. So that's the protocol here. I will not go through all of them, but basically we had 190 compounds at the beginning, for which we have more than 20,000 genes with log4 change, so with the regulation. And then developing some machine learnings like random forest, like support vector machines. We could develop some models, making some prediction and check the accuracy of the models we have developed using some test set. And here it was 86 compounds using as a test set to validate our models. So we call these tools qualitative gene expression analysis relationships in reference to QSAR. Here we are using gene expression, not structure. And that's a result we obtained with our study, where basically SVM was machine learning that seems the most performance to make a robust models. And on the validation set, we could see the accuracy was quite good if we use, so global models means all the list of genes, so the 12,000 genes. Feature selection, that's where we reduce the genes to only those who have a high deregulation. And then, if we transform the genes by 0 and 1, it was not really good. We did the same exercise using pathway information. As we know that genes were related to some pathway, we could use the pathway information. And we could see also we had some good models which were reliable to do some prediction for new compounds. So like that, we were quite happy because we can make some prediction without using the structures. And that's something that can be interesting for some pharma industry, but also chemical industry, because sometimes they don't want to give you the structure of the molecules because it's sensitive. Well, they don't want to share. But at least they can share, the readouts of the transcriptomics data, so all the genes regulation. And so with our tools, we can make some prediction without notice which compound it is, if it's toxic or not. And it gives like that also some prediction and so some collaboration with some industrials that want to test their compounds, but without giving the structure of the compounds on which they are interested to. Second point, which is interesting with such kind of tools, that's like with descriptors where you can check which structure part of a molecule seems to be important and related to some toxicity endpoint. You can do the same also with transcriptomics, but instead of the structures, you will be able to suggest what are the genes that seems to be the most involved in the accuracy of your models. So you can define some list of genes, a kind of a signature of genes that seems to be important to discriminate between a compound which is toxic, in our case DILI, or compounds which are not DILI. And what you can see looking on the gene of importance, you can do also at the pathway levels and looking to pathway which seems to be highly deregulated by DILI compounds and can be used for discrimination between DILI and non-DILI compounds. We did a similar analysis recently for compounds that have an effect on the estrogen, androgen, and thyroid receptors. And on this exercise, we use another dataset, which it's called links. And in links, you have basically more than 30,000 compounds, if I'm correct, with gene expression data measured on different cell line at different time and different dose. They make some restriction of genes. It's not a D, it's a S, sorry. But instead of using the 12,000 genes where you can measure the regulation, they consider only a small set, which is called landmark genes, which contain, 978 genes that seems to be the most important in the cell cycle of your cells. And so if you have some deregulation there, you will have some deregulation of the cell. So that's the information we use for the transcriptomics. About the compounds, we use the data from SERAP, Compara, and from literatures on two publications where we have information about compounds that show some activity on the estrogen receptors, androgen receptors, and thyroid receptors. So at the end, we can see on the table we got for two cell line, MCF7 and A549, more than 1,000 of compounds categorized as active or inactive. So we didn't use continuous value, but really a threshold that would define active or non-active on ER, IR, and TR. And then, we used several machine learnings model to check which one was the more efficient to make a good prediction. And at the end of the exercise, before that, something which is interesting when you have a lot of data and you want to have an idea about if your model or your machine learning approach will be sufficient to do some good prediction. You can use, in our case, that's gene expression, but you can do the same with structures. Making a UMAP, it's like a PCA, to look on the distribution of your chemicals in a chemical space if you use the structures, but also in the biological space if you are using genes, as it is in our case. And you can see in this case, already for ER binding and TR antagonist, you could see some cluster or group of compounds defined as active for ER, for example, which were on the same area of the map. And that means so they share already kind of similar gene signatures. So you could have an idea already about if your model will be good or not, because if you have already some cluster of compounds with the same annotation and located on the same area of a UMAP or a PCA, you can assume that you will be able to make models that will be good enough to do some good prediction for new compounds. And that's what we could see here with the ER binding, with the TR antagonist. It was less clear with the IR binding, but anyway, with the machine learning process, we could see we have some good prediction anyway. So if I come to the results with ER, IR, and TR antagonist, so from each line, that's all, the different machine learnings we play to see if we can make a good models. And we could see the balance accuracy, which is defined here on the bottom. And so when you have a balance accuracy up to 0.5, you consider that you are models which is better than making a predictions by chance. So we could see with ER, the model were quite performant when we use an algorithm which is called XGBoost or random forest, which was also quite okay. And in this case, it seems that using the MCF7 cell line was more promising to have a good prediction compared to the information we have on the other cell line, which is the I549. So actually the MCF7, it's a cell line used for breast cancer. And the F549, it's a long cell line. And so yes, for here we could see we have some good prediction with MCF7, and it was up to 0.65. It was better with the AR binding. And in this case, it was better with the cell line A549, where we were close to 0.75. And for the thyroid receptors, we have basically a similar, balance accuracy, so a similar performance if we are using, the A549 or the MCF7 cell line. In most of the case, TPOT, which is a combination of different machine learning, and XGBoost was the better. But we were quite happy about the balance accuracy, which was, at least in R and TR, up to 0.70. So that means we have 70% of good prediction. And we are close to 0.65 good prediction with the ER binding. And again, like I show with daily examples here, you can also suggest what are the genes that seems to contribute the most to the performance of your model. And we could see here, the genes DTI-T4, CCN2, CDCA3 are genes that seems importantly involved in the ER prediction and some for ER prediction. And if you are looking on the literatures, some of them were already suggested as genes that could have an impact on the estrogen receptors or androgen receptors or thyroid receptors. Okay, so what I showed you previously was how we can use transcriptomics data with some machine learnings and do some kind of QSAR methods. We use another source of data, which is high content imaging. And high content imaging, it's a way where you can visualize a cell, but also different compartments inside of the cell. So you can color, for example, the nucleus in green, the mitochondria in red, the reticulum endoplasmic in yellow, and so like that you can visualize with these dyes, the different compartments of the cells. And then, you have some techniques like cell painting, for example, which is a way of screen a lot of compounds with exposed, in this cell line, and with these dyes to visualize a little bit how it can perturb the morphology of this compartment in the cell and the cell as itself. So, like in transcriptomics or like with classical QSAR, you can have a list of what we call morphological features, which will represent, the variability, the perturbation of the different compartments of a cells due to the presence of a compounds or not. And you can measure between 1,000 and 3,000 morphological features. Which can take, the size, which can take also the granularity inside of a compartment, a texture, and so like that you will reach a matrix again with a toxicological endpoint or biological endpoint, and a lot of columns with morphological features, perturbation, so value that you can use to do some machine learning and predictions models for new compounds for which you have also such kind of morphological profiling. And so this kind of technology are more and more used now on heat discovery, on assessment of chemical-induced toxicity, on studying the mode of action, recently also on determination of point of departure, and also on drug design. I put some paper here, so you will be able to read them and see, the large, possibility and opportunity to use, such high content imaging tools to do some screening and to answer some of your questions at the cellular level. So that's an example again with DILI, but instead of using transcriptomics, this time we are using the cell painting readouts, so all these different morphological features to develop some models. And in this case, we compile, 6 different data sets with different endpoints. We put some precision, sometimes with the annotation, because sometimes you have a clear separation between compounds defined as really DILI and really not DILI. But sometimes, the thresholds to define DILI, not DILI was not too clear, so we put a must to define, it's not a clear separation. And another data set called EUROTOX, which has been developed through an IMI program, for which, many compound has been tested on rodents, and for which we could have also annotation about if they were DILI or not DILI. So with this morphological signature, again, that's the protocol. We'll go fast through it, but you get the readouts from the cell painting technology. You do some normalization, some preparation of your matrix, and with the annotation as DILI or not DILI, you can develop some machine learning models. Again, what is interesting, like the UMAP I showed you before, we can have a look in advance in a kind of heat map if there were some grouping of DILI compounds or not DILI compounds. And it was interesting to see that, maybe not so clear for you, but you can see here on the top square, the yellow square, on the left, you have more blue compared to the right one where you have more red color, which means that for each morphological feature, which is defined by each column, the blue one means you have a negative perturbation, or like a deregulation of these features for this set of components on the left. And with the red, you have a higher perturbation of the cell in presence of the compounds. And so like that, you could already, see cluster or group of compounds which were defined as DILI and seems to have a similar cell morphology signature. So you could already use such kind of signatures to predict for new compounds if they are on the same signatures, say, okay, they have a risk of DILI or not. And then, if you go further, and that's what we did with the application of machine learnings, we could also do some prediction. Not using the full set of cell morphology features, because it was bringing more noise if we use the full set of cell morphology features. But if we start to do some selection of features with high variability, then we could see that we have some accuracy which were a little bit better, because up to 0.70 for some data set. And again, one of the difficulty it's when you want to link the cell morphology with genes regulation. And so that's what it starts to be interesting, to compile information you have at the cellular levels with cell painting data, but also with at the molecular level with the transcriptomics data. Because then you can start to be able to link observations that you can see at the genes levels. So what are the genes which are deregulated by DILI compounds? And observations that you see at the cellular levels, and see, okay, which compartments seems to be the most perturbed by the compounds. And so by doing that, we were able also to cluster a group of compounds by compartments, and to suggest also, because these compounds has some genes deregulation and pathway deregulation, which was common, to make some hypotheses about compounds that were, basically have a similar mode of action at the molecular levels, and that can be observed also in different compartments at the cellular levels. That's what is represented on this figure. If you want more information, so we have a publication on it, and I will not go in detail on this one. Rapidly, just to tell you that we did a similar analysis with mutagenic compounds, where we have collected around 110 compounds for which we have some cell painting features, and so we were able to develop machine learning models with a good accuracy, because the balance accuracy was close to 0.81 on the test set, and so we are able to make some prediction of mutagenicity for new compounds if you have also cell painting that has been developed for this compound. That will be my conclusion. I hope I have not been too fast and it was clear, but basically the strength of using biological data and not only chemical structures, it's you can make relationship about the response of your cell or some gene or genes after chemical exposures, and so to make some interpretation about the mechanism of action, which something that's more difficult to do with the structure of a molecule if you don't have any information before. So it's complementary. We did some analysis also on DILI and on mutagenicity if there are some complementarity with QSAR using the chemical structure. And it was interesting that for some compounds not well predicted by using the chemical structure, so we could have a good prediction with cell painting and transcriptomics data. And on the opposite, the same. I mean, some compounds that were not well predicted with our methods, but better predict with the classical QCR approach. The weakness is preparation of the matrix because it needs a careful pre-processing protocols to limit the batch effect, the plate effect, or spurious data that you could get from this biological response. So it's not so easy compared to a structures. You have plenty of tools where you can compute chemical structure of the compounds. And then, I mean, the transcriptomics and cell painting data are also developed on specific cell line at this moment, which are essentially cancer cell line. And so derogation and perturbation that we used for our models might be a little bit different if we were using primary cell line, so it's one of the limitations. And finally, I mean, again, I mean, biological data are sensible to dose and time, and so that's something you need to consider when you are making such development to use, I mean, compounds where you know you have only a mutagenicity effect and not a too small one where you don't see anything. We are aware of it. I just put this slide showing that we are a publication on bioRxiv showing comparing, I mean, 3 cell lines at 3 different time points for the same set of compounds, how they are distributed in a UMAP, and we can clearly see it's cell line dependent and it's timeline dependent. So it's something that you have really to take in consideration when you want to develop such kind of model. And so I would like to thank, I mean, PhD and postdoc who are working on this project and the CE and PLE teams, which also contribute on some of this work. Thank you for your attention, and I'm ready for some questions.
Thank you very much, Olivier, for this very comprehensive presentation. So we have time for a question, of course. So I have the first question from Damiano. Are the matrix predefined, or in case there are companies that will be able to make matrix on demand?
So they would be predefined according of the technology they are using, and you need to have also a coherence between that. As I said, for transcriptomics, you can use, I mean, a chips array, or you can use an RNA sequencing. And so the readout will be a little bit different. We are trying to find now a way of comparing one type of data to another one, and if there is a kind of coefficient of correlation or a value to tune a little bit a value from one data set to another one, which will be compatible to do prediction for any kind of technology. But yes, the technology used needs to be similar from the modeling approach and from the prediction approach.
And you think there will be a company that can make a matrix based on, I mean, you go to a company and you ask for a matrix that will do it for you?
So usually that's the other way. We are trying to adapt our models with the technology of the company. It's, for example, what we do with BioCrop. But that's something that could be done on the other way as well. Of course, they need to have the technology. Not everybody have high contact imaging tools to perform such kind of data. Same for RNA sequencing. So yes, they need to have the technology in place for doing that. Or after, I mean, we can also collaborate with platforms that we have at the university or or some other contractors to do such kind of analysis or experimental analysis, and so we can use them for our modeling approach.
Okay, there is another question from Julia, sorry for the pronunciation. The balance accuracy for ER, AR, and TR antagonists varied from 0.6 to 0.8.
Yeah.
Why do you think that is? The model principle is the same. Is it the data availability or something else?
I don't think it's about the data availability. So there are some difference about cell line, and that can be explained. We can explain it because the genes regulation might be a little bit different from one cell line to another one. So it can have an effect and an impact on the modeling and the prediction. When you are looking on the same cell line and you see some difference like that, it's because we use a specific time point and a specific dose, which was 10 micromole, I guess. And so maybe for this specific dose, which was the same for all the compounds, you have some response which are more pronounced for ER compared to AR for some compounds. And that might explain a little bit also the difference of accuracy we could get from ER and IR modeling.
Okay. So another question from Catherine. Have you identified global gene signatures associated to toxicology? It's a general question. I mean, is there any signature that can be associated to toxicology or some toxicological endpoint?
Yeah, that's a difficult question. So you could. Again, what we do with this machine learning approach, we are trying to see a kind of general effects for a specific endpoint, which means that from 10 compounds, if you have 3 or 4 compounds which have the same cell signatures and same transcriptomic signatures, you could predict for compounds with the same signature that they will be DLE. But then you have 6 other compounds which might, have an effect using different mode of action or different cell morphology perturbation. And so you will don't have only a unique one signatures for a specific endpoint, but probably a different one. And so you will be able to make some assumption and putting some confidence score. But okay, I know my compound, if it's in the same group of these 3, 4, 5 compounds, I have a high confidence about the DLE effect related. If it's only one, then the confidence about it will be DLE because it shares a signature, but it's only for one compound, will be a more you will be able to discuss about that. Yeah.
Yeah, it's always difficult, this question of signatures. I remember when the transcriptomic arrived in the field of biology, everybody toxicology thought we could have a transcriptomic signature.
Yes, but no.
That was not the case at all.
No. That's difficult because there are plenty of mechanism of action which can be related to a toxicity endpoint, and usually you have to consider the concentration. They are not have an effect on the same concentration, so you have some limitation there. And so some effect will appear also maybe later during the time, and some in a early point. So genes that will be deregulated in one condition for one compound might be different to another one that will need more time, higher concentration to see a similar effect. So it's difficult to have a clear answer to that. Yeah.
And there is a comment from Julia who asked the former question. Thanks for your response. That is very interesting. Time considerations are very important. That's what you said, and maybe underestimated in toxicogenomic experiment. That's clear.
Yeah,
That's part
agree.
of your work actually. So
Yeah, I agree.
I understand the amount of data for processing when using different doses and different times are enormous.
Mm.
But this is informative just to look at and help understand the differences between the different types of simile.
Yes, I recommend you to read the publication on bioRxiv now because it's really interesting to see the difference of observation you can have between times and cell lines, and using the two technology, transcriptomics and cell morphology, because you at the beginnings you have the feeling you don't see the same thing, but when you try to match, I mean, the genes regulation with the cell morphology features which are highly deregulated, you can start to see there are some connection and some correlation. So it was interesting to see that according of time and cell type.
And so, Mia, I've got also some question because there's no more question in the chat. So I've some question about the origin of the data. You just pointed out
Yeah,
very clearly, and also in your conclusion concerning the cell line. So I'm
yeah,
not going to ask you the question about how do you think, what about the cell line, blah blah blah. Just, if there is there any kind of effort to try to, for example, if I address DILI, okay, I will try to build a consortium or put an open project in some, okay, on the EPA-RG, for example, and
Mm.
for kidneys on other more relevant cell line, and try to build this database in a very relevant cell line that are already used in to screen some compound, are already used that can be very informative.
Yeah. I agree with you. That's one of the limitation so far. That's we use some cell line which are cancer cell line, MCF7 and 505. They are sarcoma, and so the representation of really the effect on the primary cell line or healthy cell line in some way might be a little bit different. The thing that they use this cell line because they are easy to grow usually and stables, and so you can get answers with a kind of homogeneity that you want to have with such kind of analysis, because we know it's very sensitive to protocols, to the dose, to the times, and even, I mean, if you are reproducing the same experiments in another lab, you might see some difference, significant difference, and so the cell lines for which are using such tool now, in some ways, are conservative because they know that they will see something. There are some consortium which are starting to work on specific cell line, like as you said, I mean, EPA-RG and also on kidney cell line. I'm part of one project which is Risk Hunter, where, I mean, they are doing some cell painting on FG2 for a set of 100 of compounds at different times and different dose. And there will be also some transcriptomics data. So we will have a better interpretation of connection of observation at the cell levels and at the molecular levels. But I agree, I mean, it's done only on some specific consortia with specific and maybe small data set, even if 100 for some it will be a lot. But we know that they were trying to do a kind of similar analysis on H295R cell line, which is for steroidogenesis. And the cell line is very unstable, and the results was not exploitable. I mean, we cannot use it to do such kind of analysis because not homogeneous.
Okay, so I have one last question because we are just running out of time. Just one last question, which is I don't know if it's an interesting question, but when you put on which is very interesting, when you try to cluster your molecule, okay?
Yeah.
On slide 34, you try to cluster your molecule and you can see there is some molecules that are not related at all in terms of structure, for example.
Yeah. Yeah.
Like, for example, when you cluster amitriptyline and doxorubicin, I think.
Yes.
I
Yeah.
mean, it's very interesting because it means that, okay, for some of the, because at one time the chemists, they were saying, okay, the structure is making everything. But
Yes.
this is
And
not.
that's what we want to try to tell to the people. It's not only about the structure.
Mm.
Of course, the structures and because it has been the paradigms for decade that everything is related to the structures, and so if you have a similar structures, you should have a similar effect. And in some way, I agree, but you can, I mean, come with a compounds with a new structures and have a similar effect because it will bind also on the protein, maybe with not the same binding modes, but it will have an effect on the protein or the genes. And so if you are using these classical QSAR, you will have some difficulty to predict them because your chemical space is limited to the structures that you have on your data set. And that's what we try to go up with this biological response, because then you are not considering the structures, you are considering really the response of the cell in presence of the structures. So the structure has nothing to do with the readouts and the information we use in our modeling. But that's the reason why I think they are complementary, because that's true. For some compounds, the structures it's enough to make a good prediction, but for some others, you need to go upon the structures, and using the biological response is one way.
Okay, so we are now, it's 1:05, so I think we have to stop. So
Okay.
thank you again very much, Olivier, for this very comprehensive presentation. Thank you for the audience to ask some questions. And so I wish you a very good day, and see you next month. See you in January for the next webinar. Okay.
Thank you for your time and listening me.
Yeah, and thank you for the last message, that structure is not telling everything.
Everything. Yes.
Thank you.
Bye.
Bye-bye.